Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimate to you. In the last session, we ended up with salient features of cholesterol. In today's session, we will continue with absorption of cholesterol, where cholesterol reaches the bloodstream through lymphatics. Then comes the biosynthesis of cholesterol in sequential step. Then we will continue with regulation of cholesterol synthesis. Lastly, we will deal with cholesterol pool and cholesterol metabolism. Good morning, everyone. Let us begin the topic. Absorption of cholesterol. Cholesterol ester present in the diet is hydrolyzed by cholesterol esterase. The free cholesterol incorporated into bile salt micelle is absorbed into the mucosal cell. Absorption needs micellar formation. There is specific protein which facilitates the transport of cholesterol into the mucosal cell and from the micelle. Inside the mucosal cell, cholesterol is re-esterified and incorporated into chylomicrons. The chylomicrons reach the bloodstream through lymphatics. The dietary cholesterol reaches the liver through chylomicron remnants. Plant sterols, that is sitosterol, decrease the absorption of cholesterol. The protein designated LPC1L1 is involved in the absorption of cholesterol. The therapeutic effect of the drug ezetimibe is by interfering with the function of the protein. ABC transporter proteins, that is ABCG5 and ABCG8, constitute a dimetic unit, limiting cholesterol absorption. They promote the secretion of absorbed sterols from the intestinal epithelium back into the lumen and thus regulate the amount of cholesterol incorporated into the chylomicrons. Coming to biosynthesis of cholesterol. The major sites of synthesis of cholesterol are liver, adrenal cortex, testes, ovaries, and intestine. All nucleated cells can synthesize cholesterol, including arterial walls. Enzymes involved in the synthesis of cholesterol are partly located in the endoplasmic reticulum and partly in the cytoplasm. Coming to step 1, condensation. The acetyl CoA is provided by the ATP citrate lyase reaction as in case of fatty acid synthesis. Two molecules of acetyl CoA condense to form acetoacetyl CoA catalyzed by the cytoplasmic acetoacetyl CoA synthase. In this diagram we can see two molecules of acetyl CoA combine to form acetoacetyl CoA with the removal of CoA and addition of, in the presence of enzyme, acetoacetyl CoA synthase. The next step is production of HMG CoA. A third molecule of acetyl CoA condenses with acetoacetyl CoA in the presence of and gives beta hydroxy beta methyl glutaryl CoA. The enzyme is HMG CoA synthase. HMG CoA is present in both cytosol and mitochondria of the liver. The mitochondrial pool is used for ketogenesis, whereas cytosolic fraction used for cholesterol synthesis. In this diagram, we can see acetoacetyl CoA is converted to beta hydroxy beta methyl glutaryl CoA in the addition of acetyl CoA and removal of CoA molecule in the presence of enzyme HMG CoA synthase. Next step is committed step. The reduction of HMG CoA to mevalonate is catalyzed by HMG CoA reductase. It is a microsomal enzyme. It uses two molecules of NADPH and it is a rate limiting step. Here we can see beta hydroxy beta methyl glutaryl CoA is converted to a 6 carbon unit, that is mevalonate, with the addition of NADPH plus H plus and removal of NADP in the presence of enzyme HMG CoA reductase. Next step is production of 5 carbon units. The mevalonate is successively phosphorylated to phosphomevalonate, then pyrophosphomevalonate, then 3-phospho-5-pyrophosphomevalonate. This then undergoes decarboxylation to give a isopentenyl pyrophosphate, a 5 carbon unit. In the diagram we can see mevalonate, which is a 6 carbon unit, is converted into 5 phosphomevalonate with the addition of ATP and in the presence of kinase enzyme. Then it is converted into 5 pyrophosphomevalonate in the presence of ATP and in the presence of enzyme kinase. Then it is converted to 3-phospho 5-pyrophosphomevalonate in the with the addition of ATP and in the presence of enzyme kinase. Then this 3-phospho 5-pyrophosphomevalonate is converted into 5 isopentenyl pyrophosphate with the addition of 1 ATP and removal of 1 carbon dioxide in the presence of enzyme decarboxylase. This isopentenyl pyrophosphate is 5 carbon unit. Next step is condensation of 5 carbon units. Thus, 6 numbers of 5 carbon unit are condensed to form 30 carbon compounds, that is squalene. In summary, it is written as 5 carbon plus 5 carbon gives 10 carbon, then 10 carbon plus 5 carbon gives 15 carbon, then 15 carbon plus 15 carbon gives 30 carbon. In the diagram here we can see isopentenyl pyrophosphate is then converted into dimethyl allyl pyrophosphate in the presence of enzyme isomerase. Then the two molecules of dimethyl allyl pyrophosphate are converted into geranyl pyrophosphate in the presence of enzyme transferase. Then this geranyl pyrophosphate with isopentenyl pyrophosphate combines with combines to form farnesyl pyrophosphate in the presence of enzyme transferase. Then this farnesyl pyrophosphate plus another molecule of farnesyl pyrophosphate give squalene with the help of enzyme squalene synthase. Next step is cyclization. Squalene now undergoes oxidation by epoxidase with using molecule oxygen and NADPH to form squalene epoxide. The cyclase converts into 30 carbon lanosterol, which is the first steroid compound synthesized. Next step is cutting to the size. Next, the three additional methyl groups on the carbon atoms 4 and 14 are removed to produce zymosterol. Then the double bond migrates from 8-9 position to 5-6 position form desmosterol. Then desmosterol is present in fetal brain, but it is absent in the adult brain and it reappears in the glycomas. Then finally, the double bond in the side chain between carbon 24 and 25 is reduced by NADPH when cholesterol is formed. In the diagram we can see squalene is converted into squalene epoxide, then into lanosterol, then into zymosterol, then desmosterol and finally into cholesterol. Coming to regulation of cholesterol synthesis. Regulation and transcription. The regulatory enzyme HMG-CoA reductase. Long-term regulation involves regulation of transcription of the gene HMG-CoA reductase. When sufficient cholesterol is present in the cell, transcription of the gene for HMG reductase is reduced and cellular synthesis of cholesterol is also reduced. In the diagram, we can see the different steps. At the first step, the inactivator is cholesterol, and further enzyme CoA HMG CoA reductase step, the glucagon cortisone statins is an inactivator, whereas insulin and thyroxine are activators. When cholesterol in a diet is reduced, synthesis is increased. Cholesterol regulates the expression of HMG-CoA reductase gene and LDL receptor gene. The specific recognition sequence known as Sidol regulatory element SRE is present in the DNA. SRE binding by Sidol regulatory element binding protein SREBP is essential for the transcription of these genes. When cholesterol levels are sufficiently high, the SREBP remains as an inactive precursor. The SREBP cleavage activator protein, that is SCAP, is an intracellular cholesterol sensor. When cholesterol is decreased, SCAP escorts SREBP to two Golgi bodies. Here, two Golgi proteases S1P and S2P sequentially cleave the SREBP protein, which binds the SRE and increases the transcription of HMG-CoA reductase gene. Next step is cholesterol modification. Short-term regulation is by covalent modification of the enzyme. Cyclic LMB mediated cascade phosphorylates the enzyme which is inactive. Dephosphorylated form is active. Further, the activity of HMG-CoA reductase is also regulated by rate of degradation of enzyme protein. Insulin and thyroxine increases the activity of the HMG-CoA reductase, whereas cortisol and glucagon decrease its activity. Here in this diagram we can see HMG-CoA reduced to cholesterol with the help of enzyme HMG-CoA reductase, which is active. This active protein is converted into inactive protein with the help of enzyme reductase kinase, but further back into its active position is brought by the enzyme for protein phosphatase. The sixth regulator is drugs. Lovastatin and other statin groups of drugs are competitive inhibitors of HMG-CoA reductase. So they are used in clinical practice to decrease cholesterol blood level. Coming to cholesterol pool and cholesterol metabolism. The total body cholesterol content varies from 130 to 150 grams. LDL transports cholesterol from liver to the peripheral tissues, whereas HDL transports cholesterol from the tissues to the liver. Cells of extrahepatic tissues take up cholesterol from LDL. The free cholesterol released within the cell has the following fates. First, it can be incorporated into cell membranes, or it can be metabolized to steroid hormones, especially in adrenal cortex and gonads, or it can be esterified with saturated fatty acids and stored in the cell, or it can, or the no, the enzyme ACAT, that is acyl transferase, help in this reaction. Then it can be esterified with polyunsaturated fatty acids by the action of lecithin cholesterol acyl transferase and incorporated into HDL, and then transported and finally excreted through liver. So here ends our session. Kindly like, share and subscribe the channel. Thank you.